Mendel's first matings are what we call monohybrid matings, and these are crosses involving a single gene and two alleles for that single gene, controlling a single trait. And in the case of Mendel's first matings, his monohybrid matings, for every one of these genes, there were these two alleles, one dominant and one recessive. So I've got a picture here of pea plants, either purple or white, and purple is dominant to white. Purple is controlled by a dominant allele, white controlled by a recessive allele. I think you're familiar at this point after reading the text, but familiar with the Punnett square, but we're going to go through that in the lecture as well. After Mendel finished his work, he proposed a number of things. One, he proposed that traits like flower color or seed shape are governed by factors which we now call genes. He proposed that each individual carries two versions of these factors, and we call those versions alleles. Alleles are alternative versions of genes. He then proposed that the factors separate from each other in gamete formation, which we know is during meiosis, so that each parent donates one version to an offspring, therefore restoring the two versions in the resulting offspring. So an individual is diploid, having two copies of every gene, two copies of every chromosome on which those genes exist. The gametes are haploid. Parents donate one version to the offspring, and then the resulting offspring is diploid, having two alleles for every gene. He suggested that a true breeding strain, those that he uses in his P generation, have to contain two identical factors, and then the F1, the offspring from the P generation, carry one of each type of factor. He noticed that there was a missing trait, or one that was dominant, which was always prevalent, and one that was missing or recessive. He proposed that the expression of the missing trait must somehow be masked by the visible trait, and he called this masking dominance. So the allele for the masking trait is dominant, or he said just the version for the masking trait is dominant, and the version for the masked trait is recessive. I'm going to give you some more modern terms, not terms that he used in his paper in 1865, but the terms that we use now. Homozygous is a genotype, and it's having two identical alleles of a given gene. Heterozygous is having two different alleles of a given gene. The dominant allele is expressed in either the homozygous or the heterozygous condition, so if the dominant allele is present at all, it's the one that's expressed and it's the one that's seen. A dominant trait then is one conferred by the dominant allele. A recessive allele is expressed in only the homozygous condition, so only when there are two identical recessive alleles. And what we call a recessive trait would be one that's conferred or determined by the recessive allele. Genotype is the genetic, sorry about the typo there, genetic makeup of an organism, or the combination of alleles that are seen for a given gene. The phenotype then is the physical expression of the genotype. So for example, I have a pea here that is round. Round is dominant to wrinkled. The genotype in this case, which is indicated by the big R and little r, is heterozygous. The phenotype is the physical expression, and the phenotype is round. In this case, the genotype is homozygous recessive, and the phenotype is wrinkled. One genotype that's missing here is homozygous dominant, and that would be a round seed. But notice that I don't use the term dominant or recessive when I'm referring to the heterozygous genotype or the heterozygous individual. His experiment involved taking first two true breeding plants in the P generation, and true breeding means that through successive rounds of self-fertilization, this plant never showed any other phenotype but purple. This one as well. So self-fertilization of this plant produces produces offspring that are also purple. When those offspring are self-fertilized, they're also purple. So many generations always producing purple flowers. Another way to think about true breeding is that they are homozygous for the allele that confers the version of the trait. When he mated these two P generation plants, and whether he made purple or white female or male didn't matter. The F1 generation was always purple flowered. Something was masked, the white color was masked, and that was considered to be recessive. When he self-fertilized the F1 plants or fertilized between siblings in the F1 generation, he got a phenotypic ratio in the F2 of a 3:1 purple to white flower. And no matter which trait he used of those 7, round wrinkled seeds, yellow or green seeds, tall or dwarf plants, still he got the same outcome. The F1 generation only showed the dominant of the 2 versions of the trait, and the F2 always showed a 3:1 ratio of dominant to recessive. This is explained by the genotype, or the genetic makeup of these plants. Here we have 2 homozygous plants. The only allele that this purple flowered plant can give is the dominant allele. The only allele provided by the white flowered plant is the recessive allele, and each parent can only give one version, or one allele. So the F1 are heterozygous, and the F1 can give either big P or little p to the F2. The Punnett square displayed here shows all the possible outcomes if each one of these alleles is provided in equal numbers, which is the case. Half of the gametes from this plant will be big P, whether they're male or female gametes. Half of the gametes will be carrying the little p allele, or the recessive allele. It doesn't matter if it's the male or the female gametes. So we represent them equally on the outside of a Punnett square. And always remember that the outside of the Punnett square will only contain a single letter in each spot, because these are haploid gametes, a single copy of each gene. Then inside of the Punnett square is the combination of the potential alleles. And notice that 3 out of 4 offspring will be carrying at least one dominant allele and will be purple. Notice that there's a slightly different genotypic ratio if we think about just the genotypes. We've got a 1:2:1 ratio of homozygous dominant to heterozygous to homozygous recessive. So this explains his F1 generation displaying only the dominant trait, the F2 generation displaying the dominant to recessive in a 3:1 ratio. And the model that he proposed at the beginning of the slideshow was proposed based on what he saw here in his experiments. Now, like I said, it wasn't believed. People didn't really take what he had to say very seriously until meiosis and the segregation of alleles in meiosis 1 was seen under the microscope. Mendel proposed then the principle of segregation, which we've seen in meiosis. Each gamete carries only one allele for a gene. In this case, it's for seed shape. The alleles have segregated right away in meiosis 1. So here's a heterozygous plant, and remember, all of the cells of this plant will be heterozygous for this gene. When gametes are formed, the big R allele will separate from the little r allele in meiosis 1. That's when our homologous chromosomes separate from each other. Then there's the separation of sister chromatids, but in this case we're not looking at any crossing over or anything like that. And we see there are 2 gametes, each one carrying just one dominant allele. 2 gametes over here, each one carrying just one recessive allele. These gametes are what we're putting on the outside of this Punnett square. And then inside are the genotypes of offspring. And we're only looking at one particular gene right now. There are, of course, thousands of genes along the length of the chromosome, but we're just looking at the inheritance of a single trait, governed by a single gene with 2 alleles. I want you to take a minute and push pause on the video and answer some questions based on the presence of this Punnett square. Take a while to read the question and try to answer these 3 questions, and we'll come back to this in a minute once you push play again on your video. So you've had a chance to look through the question. In a plant the color of the leaf is controlled by a gene, and the word locus means location or a spot on a chromosome. But we're going to call this gene the D gene. One allele, big D, has dark green leaves, so that means that dark green is dominant. A plant that is homozygous recessive with the two little alleles have light green leaves, which means that light green is recessive. A true breeding dark-leaved plant is crossed with a light-leaved plant. So big D mated with little d, and the F1, which will be heterozygous, are allowed to self-pollinate. Then the predicted outcome of the F2 is sort of diagrammed here, but I haven't put in the offspring genotypes. Which of the boxes, marked 1 through 4, correspond to plants with dark leaves? E shows that this box, because they're carrying a big D allele, and this box, 1, 2, and 3, will all represent offspring with dark leaves. 2 and 3 represent plants with a heterozygous genotype, carrying big D and little d, big D and little d. True breeding means homozygous. So both boxes 1 and 4 represent true breeding or homozygous plants. Now I just want to relate this to the bigger picture. Remember that we're talking about the inheritance of a single trait. So let's say that the B gene represents or encodes fur color in a certain kind of animal. Big B is black fur, little b is white fur. An individual that's heterozygous would have black fur. Remember that there are thousands of genes along the length of the chromosome, and as we're looking at the inheritance of traits, we're just talking about the inheritance of one gene, and we're kind of ignoring all the other genes that are present in the genome, or the complete set of genes in an organism. And we'll slowly but surely get to talking about more genes, the inheritance of multiple traits, and also the fact that there are more than 2 alleles often present for a particular gene. But for right now, I really want you to understand monohybrid matings, what Mendel did, and be able to answer something like this question after reviewing the material. Our next lecture will focus on dihybrid inheritance, the inheritance of 2 traits, each one governed by a single gene with 2 alleles in the population.